好，那么大家我们差不多就可以开始今天的活动了。好的，那么各位老师、同学朋友们，大家上午、下午、晚上好，欢迎参加本期 BioCosmo 的学术讲座。那么在讲座正式开始之前呢，按照惯例，还是请允许我再向大家介绍一下我们的社群。我们是 BioCosmo 面向从事一线研究的海外高校生物学者的实名制公益性社群。主群成员仅限经过身份验证的海外各大高校生物类相关专业的在读博士生及以上学者。我们致力于建立可靠有爱的社群环境，并通过社群运营团队积极连接外部资源，赋能社群发展。散是满天星，聚为一团火。我们的目标就是凝聚散布在世界的生物之心，激发海外一线华人生物社群的能量，连接优质的学术和植发资源，助力生物人的成长，同时为中国生物研究的发展创造价值。那么右下角呢，是我们公众号的二维码，还没有关注的朋友，现在可以扫码进行关注。那么在讲座的最后呢，我们还会再附上一遍我们这个小助手的二维码，大家可以通过添加小助手，成功验证身份之后呢，就可以加入我们的社群了。好，接下来就进入我们的正式活动。今天我们非常荣幸邀请到了印第安纳的王新丹教授。王教授本科毕业于上海交通大学生物技术专业，之后前往英国牛津大学攻读生物化学博士。在取得博士学位之后呢，王教授来到了美国哈佛大学医学院进行了博士后的训练，并于2017年拿到了教职，加入了印第安纳大学。截至目前，他已经在包括 Nature Communication、Molecular Cell 等著名刊物上发表了数十篇文章，并获得了 Indiana University Outstanding Junior Faculty Award、Indiana University Emerging Equity Fund for Research Award、Indiana University Trustees Teaching Award 等诸多奖项。王教授你好，感谢接受我们的邀请参加今天的活动。
谢谢冯源和一帆组织这个活动我感觉很荣幸能参加。
好的，好的，感谢您。那么我们的第一个环节呢，是想您能分享一下您在这个职业发展过程中的一些经历。那么根据您的 CV 上看呢，其实您在去到 Harvard Medical School 之前，是有先留在 Oxford 做了2年的博后，之后您在 Harvard Medical School 的博后是从2009年到2017年，那么可以说前后加起来是有大概10年的博后经历。您在这个相对比较长的时间里，这个心路历程是怎么样的呢？有没有遇到什么样的挑战？最终又是怎么克服的呢？
好，我在英国读的博士，在英国读博士，它的那个时间很短，因为是至少在我申请读博士的那时候，我进到英国的实验室，然后直接就加入一个实验室，不需要做 rotation。直接加入实验室，也不用做助教TA 什么的，所以那个博士的时间很短。其实我从加到实验室到提交论文，然后拿到那博士学位时候，只用了3年时间。3年之后，然后我的毕业典礼是在第2年的5月份举行的，所以看起来我博士大概读了4年，其实3年完成。完成之后，我就觉得东西都。那个在英国很正常，3年就是博士学位，咱一般人多一点时间完成。我拿到学位之后，觉得好多的课题都没有完成，然后在美国读博士的同学，一般他们都要读6年，我就感觉好像是受这个训练比较短，所以我在同一个实验室又待了2年半到3年的时间，那个就总共加起来，在我博士的实验室一共待了将近6年的时间。所以那个跟在美国读博士的时间差不多，然后我就想做一个博士后，要去美国机会更多一点，然后就到了美国哈佛大学做博士后。做博士后的时候，我也想大概3、4年时间 博士后，然后就找工作呗。结果在3、4年的时候，正好 Illumina sequencing，突然就兴起了，而且在一般的实验室都能够付得起这个深度测序的这种实验刚刚兴起。然后我就有3个实验需要用到这个深度测序这种技术，我就想把这几个实验做到我的物种里边。这段时间一共花了3年，需要做很多的优化，经过很多挫折，然后最后终于做出来了。做出来之后就感觉哇，一下子有很多的问题就可以用这些技术来解决。所以我第2次在哈佛做那个博士后也做了很长时间，中间就因为要让把一些技术做出来，然后我感觉这些失败或者那些挫折最后还是挺值得的，因为确实是对我以后的研究提供了很多方法。所以主要的心路历程就是说什么呢？我觉得我最重要的还是喜欢做实验，做那实验就感觉挺高兴的，然后即使是失败了，也没有感觉特别的艰难，艰难的时候也感觉能过去，所以感觉是不知道是把自己的爱好当成了工作，还是把工作当成了爱好，反正就是也没有感觉那么长。回头想想是很长的，从读博到得到教职，一共是13年的时间。回头想想时间很长，但经历的时候没有感觉时间那么长。
可以，这大概就是热爱吧。那么相当于是您在这个博后已经做了3、4年的时候，这个next gen sequencing 开始突然就rapidly develop。那么是您在这个决定在这个做了3、4年之后，新开一个题是什么样的，或者是新进入用一个新的技术来用到您的这个模式生物里面，这个在做这个决定的时候，您有什么遇到什么挣扎的地方呢？因为毕竟你是已经做了一段时间，然后接下来可以预见的又是一段很长的时间。您在做这个 decision 的时候，有遇到什么心理上的这个障碍需要克服的吗？
是，我觉得现在咱们都是做生物的啊，做生物的有的时候你想一个课题，想做一些东西，他那个想法并不是限制你做不做出来的一些决定因素，最重要的因素还是你有什么技术。我在做博士后，在哈佛大学做博士后大概2、3年的时候，我那个用到的主要的技术还是以前我用过的一些技术，比如说看显微镜、荧光蛋白啊，然后做细菌表达那个基因表达了什么的，都是以前的技术，确实还能优化。但一旦出现了新的技术的时候，我就发现如果能把这些新的技术用进来的话，能给我的那个实验方向增加很多可能性。所以去优化一个新技术在一个物种里边其实是挺难的，但是你要想想它的付出也有回报，所以没有太多挣扎。当时的时候也没有想需要花那么多时间，觉得别人都做出来了，我也试试呗，我也能做出来。但是一做的时候发现换一个新的物种，在每一个很小的细节都需要做很小的调试。你要是调试的差不多，它就是不是非常的 reproducible， 就是这次做出来，下次做不出来，就比较令人还是比较抓狂。所以你这个调试之后，让它每次都能做出来，确实是花了很多时间。但我就像我刚才说的，所有的艰苦都是值得。
是，您最后也是那个成果应该是发到了 Nature 对吧？所以确实一个新技术，然后结合新物种，这些东西都是，然后再进行一个不断的调试，一个长时间的训练，这都是 怎么说一个成功必不可少的一个要素吧。那么下一个问题，在您博后出站的时候，您有考虑过业界的工作吗？因为现在很多人啊，大多数很多博后他都会这个逃离这个学术界，跑到业界去。那么您是在当时是为什么会选择了这个学术界呢？
当时我跟你说，我学了这几个新技术，然后突然间就有很多课题可以做，然后我就跟我博士后的导师就说，我想试试，有自己的实验室，然后领导一帮学生跟我一起做，就是因为到那个节骨眼上，我就是没有想过去工业去做报 industry 里边，就是在那个时间点，当时就是那么想的。然后我跟我博士后的导师说，我当时还想他可能会说你现在做的都做到一半，你想不想把它做完了再走啊之类的。结果我的博士后的导师他恰恰相反，他说行啊，你有很多没做完的东西，你到时候成立一个实验室自己做嘛，都带走。然后我就觉得那就试试吧，然后我就申请了就是就觉得试试吧，就申请了那个当时也是印第安纳大学在招人，我就申请了，这是我递的第一份申请。然后申请之后，觉得材料都准备好了，那就再多申几个吧。所以一共就申请了6个地方，一共有2个地方给我 offer， 然后最后我来到了我现在这个印第安纳大学，主要有2个原因。一个是我的现在的同事里边做细菌方面的同事特别多，然后感觉会有很多的合作机会。第二个就是我有2个朋友，在我读博士后的时候，他们也在那儿做博士后，他们2个都是在印第安纳大学不同时间段毕业的博士学位。我觉得哇，这两个朋友都这么优秀，都是在印第安纳大学读的博士。如果我去印第安纳大学的话，能招到他们这样的学生，那我就太牛了。所以就这样我就来了这儿结果真是来了这儿之后有很多的合作机会，然后招到了非常非常优秀学生。
是，可以，的确，其实 IU 在这个传统的 genetics 方面是有一定优势的。您最后也选择了加入 IU，然后到学术界里面应该是很符合您一贯以来一个期望了。那么下一个问题，其实恭喜王教授我们知道您在今年也是成功拿到了 tenure 那么从这个刚拿到教职，然后到 tenure 这个过程中，您有没有什么成功的经验可以和我们一起分享一下呢？
这有点老生常谈，从教职到拿到那个终身教授，最重要的就是无非就2点呗，发文章，然后就是写 grant。我在刚来的时候，学院那边就给我安排了2个比我更有经验的导师当我的导师。刚进去的 junior faculty 都有一些 mentor。这时候这2个 mentor 他给我的建议就是，学校给了你很多的那个 startup， 你要把它用掉，你要先集中精力去做实验。去把你的实验室弄好，去积累一些 preliminary data， 积累一些那些原始的数据，然后再去申请 grant。这是他们2个给我的建议，然后我觉得可以，所以我在刚开始3年的时候就没有写过任何大的 grant， 小的一些写了一些很短的那些，我觉得不怎么算。那些大的 grant 我就根本就没写，我的 mentor 也觉得对，就这么做。所以我在感觉积累了很多数据的时候，一写 grant 的第1次申请就成功了。这个就是我感觉对我帮助比较大的，就是我用了比较适合我的这个建议。然后我也有很多其他的同事，他们有不同的 mentor， 那些 mentor 可能就会建议你一进来的时候就可以申请 grant。每个人申请 grant 的时候，有可能都会有几次的失败，最后才能成功。然后也有同事是用那种途径，最后也拿到tenure。所以真是不一定说哪种 更好。你要是去觉得哪个建议更适合你，对我是另外一种。刚开始的3年就主要用在研究上面，到时候有了数据，自然申请 grant 就容易一些。
没错，没错，这是一条非常好的选择。那么您在这个初创您的 lab 的时候，您当时有学生吗？还是您就是 postdoc 和您自己在产出这个preliminary data？
我刚开始来的时候是1月份来的时候没有学生，然后就实验室，而且我的实验室还没有那个实验室那个 space 实验室那个屋子还没有被准备好，所以我刚开始的几个月时间是在其他的实验室里边，别人借我一个bench，我就用他们的各种仪器啊试剂，感觉很开心。在别的实验室，我是自己一个人做了很多实验。然后等我的实验室都准备好了之后，我就下到我的实验室，然后开始买仪器啦 set up。到招生的时候，那年8月份我也没有招到学生，这个有很多个人原因，家里边有些个人原因，在招生的时候我没在这，所以也没招到学生。所以从8月份到下面一个1月份，就相当于从入职到整整的1年时间，实验室没有其他的人。然后在第2年春天的时候，有一个本科生想要跟我一起做研究，就是有这个本科生跟我一起做了5个月的时间。然后我到第2年招到了学生，然后招到了技术员，慢慢的才是把实验室建起来。刚开始那段时间一个人还是挺说有点孤单有点孤单，但是挺不一样的经历，不一定每个人都这样经历，但是之后去想想也没有那么可怕。
那么其实 generally speaking， 是不是刚新进的 PI 在开始的时候自己其实是要干大量的活的？
是我在1年时间没有其他的人的时候，肯定都是我干所有的活儿，连准备那些试剂啦，什么把东西放哪儿啦，什么autoclave 那些 waste 什么都是我自己做的。即使是在我前3年的时间，我没有去 grant， 然后 teaching 的 load 也不怎么大，我在多数的时间都在 bench 上面干活。但现在，即使是现在偶尔在不用做很多 teaching 的时候，我也更喜欢在 bench 上面。是每个人都不一样，像我的同事，有的已经 full professor 很多年了，他们还是在 bench 上干活，但是有的同事就更喜欢写 grant 了什么的，也许是也没有时间去在bench上干活。所以我觉得我可能只要有时间，我还是愿意去做一些实验。
可以，也是因为可能您这么努力，所以您现在的 lab 规模也是相当壮大了，恭喜恭喜。那么看这个时间呢，其实我们还有允许再问最后一个问题，就是说在您这个整个的职业发展过程中，从一个学生到一个 scientist， 您觉得有没有一个事或者一个人对您这个影响非常深远的给你带来很长久的一个影响？
我觉得有一个事，也有一个人。这个事吧，就是在我申请去外国读博士的时候，那年我大4递交申请，然后第2年春天在等 offer 的时候，正好就赶上那个非典。这个非典就影响了我们整个这一届毕业然后想出国的学生，因为我们申请的多数都是美国的学校，我申请了20多个美国的学校，然后我是一个 offer 也没有拿到，我的有好多朋友拿到了 offer 也没拿到签证，所以很多朋友我一届的朋友都是第2年才来到美国， 他们去了霍普金斯大学呀、密歇根大学，最后都去了挺好的大学。我觉得这个非典还是改变了我们人生的轨迹。如果是没有非典的话，我去哪儿真是不一定。然后去那个英国读，这件事儿改变对我的轨迹改变挺大的。去英国也是一个非常偶然的事情。我跟你说，我在申请的时候，申请都是美国的学校，我写了一个 list， 英国的学校我也想申请。然后申到第一所学校，申到一半的时候，发现人家英语成绩要雅思，我考的是托福和 GRE， 我也没有成绩。就想英国学校就别申了吧。那第一所学校已经申到一半了，而且这学校也不要申请费，那我就填到底吧。我就把这个申请就填了之后，我就相当于差不多就彻底把它给忘了。忘了之后到3月份，第2年3月份也没有拿到 offer， 然后一封都是拒信。我看这怎么英国也来了？我怎么不记得英国申请了？我就看是牛津大学，发现对，第一所学校我确实申了，那拒信吧，一打开一看是一个 offer， 然后就感觉哇，这个非常非常偶然，然后我就去了呗。我感觉也是非常非常的幸运，这个英国这个大学牛津大学这个导师，他那时候已经是功成名就了。然后可能招学生不知道他是怎么就把我给选去了，而且直接就去了，也从来没有面试过，也没有打电话，没什么，offer 来了我就去了。我觉得我博士的导师把我招到他的实验室，是彻底改变了我这个以后的科研的道路。像现在我做的科研的课题，还有一些习惯啦、什么想法啦、什么。都是跟他有关。这个我的博士导师就是改变我轨迹的一个非常重要的一个人。
可以，所以这个什么塞翁失马焉知非福呀？你美国那些学校去了，最后你随便一声拿到了一个 Oxford 的 offer， 这是对非常不错的机遇。
幸运。
可以，那非常感谢王教授的回答，实在是受益匪浅。那么接下来呢，就可以进入我们今天的这个正式的学术讲座环节了。大家有什么问题的话呢，就可以打在我们的 chat 聊天框里，我们会在最后的 Q&A session 提问王老师。那么 Dr. Wang, please feel free to take over and start sharing.
Okay, next, I will talk about my research in English. I really don't know how to talk about it in Chinese. My lab studies the chromosome organization, and we use the bacteria as a model organism. So the question is, why do we study bacterial chromosome? So here is a EM, electron microscopy image. So this is a gently lysed bacteria cell, and this is the chromosome. So the chromosome DNA need to be compacted about more than 1,000-fold inside the cell. And in human cells, the DNA is even more compacted. So we want to understand how this large molecule is organized inside the bacterial cell. So why do we care about the chromosome? The chromosome contains all the genetic information to define a species. Why is a bacterium a bacterium? And why is an elephant an elephant? It's all encoded in the sequence of the chromosome. And we want to understand how this molecule is organized. And then why do we use bacteria? So bacteria are tiny. But bacteria is one domain of life, and it's just vastly on the earth everywhere. So understanding one domain of life is interesting itself. However, all the species from bacteria to humans they have common mechanisms. And we want to use that bacteria as a tool to understand the common mechanism of genome organization. And some bacteria pathogens, and we want to understand whether there are unique features of these bacteria that can be exploited for therapeutic interventions. So this is why we use bacteria to study the chromosome. So just some very general information. This is a bacteria cell, and most bacteria has a single circular chromosome here, and it has one replication origin. During replication, replication is initiated at the origin, proceeds bidirectionally. Eventually, the molecule is replicated and segregated, cell divide, and there are two daughter cells. And this DNA molecule is large, but I just draw it as a circle here. About 90% of bacteria have only one chromosome. But there are 10% of bacteria contain the so-called multipartite genome. So they have more than one chromosome. It can be circular, it can be linear, it can be chromosomes, it can be plasmid. Some species contain more than 20 different replicon. We call each of these molecule a replicon. Some species have more than 20 of them. So these are multipartite genome, and they are mostly seen in pathogens or symbionts. Having multiple replicons may be important for the pathogenesis and the survival of these species. And but having multiple pieces is challenging for their organization. We want to understand how these different molecules are organized. So the topic I will be talking about today is the organization of a multipartite bacterial genome. It's a bacterial genome that contains different pieces. All the work I will be presenting was done by two very talented graduate students in my lab, Zhongqing Ren and Qin Liao. They are truly amazing students. And the species I'm going to talk about is called Agrobacterium tumefaciens. So this is a new species, and this is a species new to me. When I joined IU, my colleague studies agrobacterium for a different topic. And I found that, oh, it has 4 parts of the genome. I want to look at it. And my student was very brave, and they took over. So we want to understand how this genome is organized. It has a circular chromosome called chromosome 1, a linear chromosome called chromosome 2, and 2 very large plasmids. Chromosome 1 is called the primary replicon, and chromosome 2 is the secondary replicon. These plasmids are also secondary replicons. So we want to understand how are these different molecules folded in the cell, and how do they interact with each other. To address these questions, we use this approach called chromosome conformation capture. So this is a technique that uses deep sequencing to understand which piece of DNA is interacting with another piece. So it's looking at a genome-wide scale. So this was one of the techniques I adapted when I was in my postdoc lab. So here is how it works. We grow up the cells, then we add a cross-linker called formaldehyde to rapidly fix everything. If 2 pieces of DNA are close to each other, these will be captured. So this is the capture part. So then we gently lyse the cell and use a restriction enzyme to cut the DNA into small pieces. Then we dilute and lyse it a lot so that the different pieces are not going to randomly collide to each other to open. But the pieces that are cross-linked, those will be together all the time. Then the ends, we will next ligate the ends to join them, and then share the DNA into pieces and sequence everything. So if during our sequencing, we do paired end sequencing, if the two ends go to the same piece, we throw those data out. But if the two ends belong to different pieces, then we say, oh, these two things are interacting with each other. So we sequence everything and find which piece of DNA is interacting with another piece. The end result is called a high-C contact map. So I'm going to first show you an example using this species Bacillus subtilis. It has one circular chromosome, about 4,000 kilobase pair in length. The high-C result is all in this picture. So basically the chromosome is, the whole chromosome sequence is put on both the X and the Y axis. Every dot on this map has a XY location, and it has a color. The color of this dot tells us the interacting frequency between these two locations. So it's a 2D map, and we see that the strongest interactions is on the primary diagonal. This primary diagonal X equals to Y, and the texture of this primary diagonal means that the tells us the domains of this entire molecule, the local interactions along the entire chromosome. Then the next feature that is very prominent is this secondary diagonal. When you trace the interaction partners, you'll see that these interactions belong to the opposite chromosome arm. And these two arms are basically, the circular chromosome is not folded as a circle, but is interacting, the two arms are interacting from the origin to the terminus, generating these zipper-like interactions. So we found that Bacillus genome is folded like this. There are other features, but in general, the two arms are folded like this. In recent years, high-C was adapted to multiple species. And in most bacterial species, we see this X-shaped high-C pattern. And basically, it means the arms are linked, interacting like this. E. coli is an outlier here. E. coli doesn't have these inter-arm interactions. And so after much study, people found that there are two major differences between these two kind of patterns. First, in Bacillus and other species, we see this X-shaped pattern and its inter-arm interactions. It has two important factors. The first one is a protein called PAR-B. It's a centromeric protein of the chromosome. You can see, just for simplicity, you can consider that this PAR-B protein binds to some sequence near the origin and form the bacterial centromere. This is one thing. And then SMC is another protein. It's conserved from bacteria to humans. SMC is loaded to the chromosome and then moves from one end of the chromosome to the other end. So this loading and movement generate these inter-arm interactions. In E. coli, it doesn't have this partitioning protein. And the SMC complex is a different family. So they are not loaded to the origin region, but it's loaded randomly. So eventually the E. coli chromosome forms like a donut and not like a zip. So these two proteins will come back in the next few slides. Okay, so now we see these two kind of patterns. Some bacteria has this X-shaped pattern. Some bacteria is more like a donut-shaped chromosome. What happens to Agrobacterium tumefaciens? It has 4 parts. It has a circular and linear chromosome. What's going on in there? So my student Warren Zhongqing did this experiment, and So here we will be looking at the high-C map of agrobacterium tumor patients, right? It has 4 parts of the genome. Let's first look at the chromosome one by one. Chromosome 1 has this X-shaped pattern. It means that this chromosome has inter-arm interactions. Chromosome 2 is a linear chromosome, and it also has this X-shaped pattern. So the linear chromosome is also having these inter-arm interactions. Now the 2 plasmids, they are smaller. If we zoom in and look very carefully, they do not have inter-arm interactions. Okay, these are the 4 different parts. They organize differently. Apparently, the topology of the molecule doesn't matter. It seems like the chromosomes are organized differently. Next, what's the interaction between these different molecules? The interactions between chromosome 1 and chromosome 2 can be found here and there. So by now, you have figured that this map is mirrored about the primary diagonal because the interaction between X and Y is the same as the interaction between Y and X. So this map is mirrored. We see that the chromosome interactions between chromosome 1 and 2, the most strongest interaction is this black dot. If we look at the interaction partners, these are the interactions between the 2 replication origins. So the origins of the 2 chromosomes are touching each other. And we also see these X-shaped interactions right here. So if we trace one sector of the X, basically it's one arm of chromosome 1 is interacting with one arm of chromosome 2. And now there are 4 sectors. Together, this shaped pattern means that both arms of chromosome 1 are interacting with both arms of the chromosome 2. Basically, these 2 chromosomes are also aligned from one end to the other. Next, the rest of the map tells us the interactions between the chromosomes and the plasmids. And basically, we see more dots right there and more dots here. So these dots means that all the origins of the 4 replicons are interacting with each other in this shaped pattern. So from there, we know that this is how this complex genome is organized. Now the question is, what is mediating these interactions, right? So the first is, let's look within each chromosome. Chromosome 1 has inter-arm interactions, chromosome 2 has inter-arm interactions. So what's mediating these interactions? Just now, I mentioned the 2 factors. Part B, you can think is the centromeric protein. It binds to the origin region and it loads SMC. SMC encircles the DNA and moves from origin to terminus, generating these inter-arm interactions. So we wondered, is SMC doing this inter-arm interaction for both chromosomes? So Zhongqing deleted the SMC protein from this organism, and we found that indeed, when SMC is gone, these inter-arm interactions on both chromosomes disappeared. So indeed, SMC is required for these inter-arm interactions on both chromosomes. Next is the loader. So part B is the loader. For other bacterium, part B is the loader. What is loading SMC to the 2 chromosomes? Okay, what is the loader here? So on chromosome 1, there is a part B homologue called part B1. And we wondered whether part B1 could be the loader for SMC on chromosome 1. Part B1 is essential, we can't delete it. So Zhongqing made a depletion spring. When part B1 is absent, we see that on chromosome 1, inter-arm interactions disappeared. But inter-arm interactions on chromosome 2 is still there. So part B1 is the SMC loader on chromosome 1. but not on chromosome 2. So what could be the loader for chromosome 2? Yeah, so on chromosome 2, there is another protein called RAFB. It's a distant homologue of part B. But these RAFB homologues are also present on the plasmid. So we wondered whether RAFB could be the SMC loader on chromosome 2. And if it is the loader, why is it not loading SMC to these other things? So RAFB turned out to be not essential, and we could delete it. So when Zhongqing deleted RAFB, we found that the interarm interactions on chromosome 2 is still there. So RAFB is not the SMC loader. And that makes sense because RAFB is not loading SMC to the plasmids anyway. So what is the loading factor for SMC on chromosome 2? And this is a big question, and we are still working on that. So now we know the interaction and the factors within each chromosome 1 and the chromosome 2. The next question is, what is mediating the interactions between the replicons, right? So it turns out that when we were building these mutants, we already had some answers. So when RAFB was deleted from the chromosome, we see that there was no change on interarm interactions on chromosome 2, but there are other striking changes. So in the red box is the interaction between chromosome 2 and the rest of the genome. We see that in the wild type situation, chromosome 2 is interacting with all the replicons in the origin region, and the arms of the 2 chromosomes are aligned. But in the absence of RAFB, we see that all those interactions disappeared. So without the centromeric protein, chromosome 2 is like a singled out. It's not interacting with anybody else anymore. But an important thing is the interaction between chromosome 1 and the plasmids are still there. So the centromeric protein is required for this chromosome to be grouped with the other replicons. Okay, so then when we depleted the part B1 protein, we see first, similarly, chromosome 1 is not interacting with other replicons anymore. But chromosome 2 is also not yet. The interaction between chromosome 2 and the plasmids are also gone. So without the centromere of chromosome 1, all the interactions fell apart. So chromosome 1 centromere seem to serve as the hub for these different replicons to be grouped together. Okay, so then here is what we found so far. Now the next question is, how is this genome oriented in the bacterial cell? And to do this, we optimized the visualization system. Basically, 2 of my students, Zhongqing and Qin, labeled the replication origin of each replicon. And they found that they are all labeled in green, and we look at them one by one. So we see that, for example, for chromosome 1, the origin is localized to the cell pole. After replication, there are 2 copies of the origins, and they are localized to both poles. And it's the same case for all the origins of the replicons. So from there, we concluded that the genome is clustered with the origin at the pole, and after replication, they localize to both poles. Now the question is, what happens in between? How do they move from one copy to the other? How do they move there? So to do this, we did a time-lapse microscopy to follow the origin movement with time. So we label the origin of chromosome 1 green and the chromosome 2 in red. In a newborn cell, the two origins are co-localized to the cell pole. It's here, it's yellow in here. And during replication, chromosome 1 replicates first, and one copy stays, the other move to the other pole. But very curiously, the unreplicated copy of the second chromosome, this red one, travels with the moving copy of ORI1. So when this red focus is outside of the cell pole, they get replicated and segregated, and eventually both copies go to the pole. Okay, the model is summarized here. When we co-localize ORI1 with the plasmid origin, we see essentially the same phenomenon. So the data are now summarized in this model here. In this agrobacterium has 4 parts of the genome. It's organized like this. During replication, the origin of chromosome 1 replicates first, and one copy moves away. As it's moving, the unreplicated smaller replicons travels a little bit with this green origin, and then they get replicated and segregated. So we later in a follow-up study, we found that origin clustering promotes genome maintenance and regulates DNA replication. So this is the first part of my story, and but next I want to tell you the backstory of this story. So just now I show you this Hi-C map, and the things seem to make sense. It's complicated, but we can make sense of it. But this was not the first time we didn't get this result. The first time we did Hi-C in agrobacterium, we got this result. It was really confusing, really puzzling. So for example, in green box, this is chromosome 1. And chromosome 1, there seemed to be something strange right here. And here is chromosome 2, right? Looks normal, but then it's just very weird. It looks very, very puzzling. And we were scratching our head and just couldn't really make sense of it. It's just too complex. Why is it very hard, so hard? So, but it really looked like the chromosome is broken somehow. It seemed to be not connected. So chromosomes are large molecules. We expect the interactions to be continuous, but this is not the case. So really, out of desperation, we took a pair of scissors, cut this part out, and then just cut the map and their junctions into small pieces. Then we just like solving a puzzle, shuffle the different pieces and see, oh, eventually we got a map that can be interpreted and we can make sense of. So we did more experiments and finally found that the strain, the wild type strain we got for our Hi-C experiment has a genome rearrangement. So the 2 chromosomes are fused into one large chromosome. So they are broken at a specific location and then rejoined and form this dicentric chromosome, a chromosome that has 2 centromeres and 2 replication origins. We call this the fusion strain. And the other wild type strain I showed you, we call them the original strain. They are supposed to be the same thing, and they behave very similarly. Only when we look at the genome, we see that this isolate is not normal. Then we want to say, oh, did this fusion event happen in our lab? So we requested wild type strains from about 20 labs, and 12 of them has this original strain, and 8 of them have the fusion strain. Then we asked them, where did you get this strain, and track the different publications. Eventually, we found that the fusion event likely happened in Steve Farran's lab around 1986. And then this strain, the wild type strain, get distributed as the wild type, and without knowingly, they have this fusion. And we did some tests and found that these two strains just behave it's like exactly the same. If we didn't know that this is a fusion, it has a fusion chromosome. The fusion strain has no detectable defects at all. So for people who study metabolism or who study cell attachment, for many, many different studies, it doesn't matter which strain you use. I guess they behave the same. But we are interested in the genome. So this is like something we are very curious. This chromosome has 2 centromeres. Are these 2 centromeres both functional? Most bacteria has only 1 replication origin, but this guy has 2. Are both origins working? So we want to test, do some more tests and just follow our curiosity. So the first question is, are both origins firing? Are those both origins be used for replication? And to test this, we use another method that combines, uses deep sequencing. We grow both populations to exponential phase, and not just get the cell, get the DNA and sequence everything to do so-called macro frequency analysis. Basically, this is the genome replicating profile. On the x-axis is the entire genome. We have chromosome 1, chromosome 2, and the 2 plasmid. On the y-axis is basically the number of reads, and it's a relative copy number. We see that for each replicon is a triangle shape. For chromosome 1, this is replication origin. In the population, origin replicate first, and we have more copies. Terminus replicate last. We have lower copies, so the profile is like this. This is for chromosome 1. For chromosome 2, the height of the peak is lower. It means that chromosome 1 replicate first, chromosome 2 replicate later. And this is consistent with our microscopy data. This is for the original strain. For the fusion strain, when we did the same, we see that both origins are firing. And if we calculate the relative frequency of the firing, it's exactly the same. So these 2 origins maintained the same replication program as the original strain. So yeah, both origins are working. Now the question is, are they really needed? We have a bacterial chromosome. This guy has a linear chromosome. It has only 1 origin. Now, can we try to delete one or the other? And these have 2 centromeres. Can we remove one of them? So we at the beginning, we are just trying to delete things and remove things one by one, and very hard. But when we can't achieve the mutant, does it really mean that it's essential? And there are just too many things to test. So then we developed this. Okay, the question is, are both origins needed and are both centromeres needed? And we developed a screen, genetic screen, to do it systematically. So basically, we get our cells and use a transposon to generate a mutant library. This transposon will hop into the genome or hop around, and they can insert into the genes that are not essential. When they are, then we harvest all the library, okay? And then we just sequence it all to find where the transposon is inserted. If it's inserted in an essential gene, the cell will be dead. We won't recover the mutant, and we won't have a transposon insertion in there. So, so we did this TNSeq, transposon sequencing. The genes that do not tolerate transposon insertions are essential genes. So let me show you a stretch of this genome. We here on the x-axis, we have a genome location. These are different genes. So each red bar here is a position for transposon to be inserted. And the height of the bar tells us how many insertions are there. And we see that in this stretch of genes, there are transposon insertions. So these genes are not essential. Okay. Next, now in the next slide, you'll see that now I will have two tracks. On the first track is the original strain, and on the bottom track is the fusion strain. We now look at one stretch of genes, and we see that this gene, DNA A, doesn't tolerate transposon insertion. And it is the case for both the original strain and the fusion strain. So DNA A is essential for both strains. And DNA A is required for the replication of chromosome 1 origin. So we now know that we can't delete DNA A. Then we looked at all the elements that are required for the replication and segregation of both chromosomes. Just now, I showed you that DNA A is essential. Here is the DNA sequence that is the replication origin of chromosome 1. Both strains do not tolerate transposon insertions. So here is the partitioning locus, and you can consider this is the centromere proteins. In both strains, they do not tolerate transposon insertions. And similarly, for chromosome 2, the origin, replication origin, the origin initiator, or the segregation machinery, they are all essential. So from here, we concluded that the replication origins, both of them are essential in the fusion strain, and both centromeres are also essential for the fusion strain. We tried to delete it, we couldn't, and now using this screen, we can see it quite clearly. So we found that all the essential genes in the original strain are still essential in the fusion strain. The next question is, are there genes that are not essential in the original strain now become essential in the fusion strain? And we looked, and indeed, there are two of these genes. It's called XERC and XERD. We see that in the original strain, there are transposon insertions in these genes, meaning that deleting these genes, the cells are fine. But in the fusion strain, there are no transposon insertions. Then we want to validate, is it really the case? We try to delete them, couldn't, so we made a depletion strain. In the presence of inducers, proteins are there. Without inducers, the proteins are gone. And we found that in the fusion strain, when XERC or XERD are depleted, the cells do not form colonies. So indeed, XERCD are usually not essential genes, now become essential in our fusion strain. So these proteins have been studied very well. And what do they do? For bacteria that have a circular chromosome, in the newborn cell, they have one chromosome. After replication, they have two circles, and the cells will divide. They generate two cells identical to this previous generation. But occasionally, during replication, there might be homologous recombination, and then it generates this dimer chromosome, this dimeric chromosome. When this is generated, cell division cannot complete. The cells are in trouble. And in circular chromosome, it expresses this protein called XERC and XERD. It binds to a sequence, this blue sequence called the deep site. And it cuts the DNA and rejoins them to resolve the dimer into monomers. So for bacteria with a circular chromosome, in every cell doubling, about 7% of bacteria will form this dimer chromosome. So in the entire population, about 14% of the cells will go through this. And if the cells have XERC and D, they will be resolved and cells are totally fine. If the cells do not have X, E, R, C, and D, there will be some cells dead. But in general, the vast majority of the cells will survive. So these genes are important, but not essential. And it's for circular chromosomes. Now, in our case, in the original strain, we have the circular chromosome. It has a deep side, and there's a triangle here. And XERC and XERD will work on the deep side, but they are not essential, as I just showed. For this dicentric chromosome, the deep side is in between the two origins. Why do they become essential here? Okay, so this is my last slide, and this is our model of why it is essential in this fusion strain. Okay, in the original strain, there are two chromosomes, and at the replication, RA1 replicate first, RA2 replicate later. Eventually, there will be two copies of chromosome 1 and two copies of chromosome 2. And each copy of each chromosome needs to move to the opposite end of the cell. And for chromosome 1 and chromosome 2, each has two copies. It doesn't matter whether how these two copies move, the directionality doesn't matter. So after the segregation, each daughter cell will receive one copy of each, and there will be no problem. Just now I showed that for the fusion strain, the two origins are still essential, the two centromeres are still essential, and they remained the same replication and the segregation profile, right? They remained the same program. So after replication, they segregate. Now there can be an issue because these two origins, the red and green, are now connected to the same molecule. If this one copy of RA1 and RA2 belong to the same chromosome, if it goes to the same direction, then there should be no issue. However, if this red and green go to opposite cell poles, there is a problem, okay? Because now these molecules are blocking cell division right here. If this block is not resolved, in every cell generation, 50% of the cells will be eliminated. And this entire progeny will be dead. But luckily, in our case, the deep side is right in between the two origins. And XERC and XERD will find the deep side, cut and rejoin the molecules, and resolve this situation to make cell division progress normally. So that's why we think XERC and XERD become essential in our fusion strain. So now the conclusion is dicentric chromosome can be stably maintained as long as it has a dedicated resolution system. So from there, this is my acknowledgement. I'm very grateful for my small lab. And here, this Agrobacterium cumafaciens, this species was a new species I adapted. Is because of my colleague here at Indiana University, Clay Fuqua. Ian Button is his graduate student, and they really helped us to learn all the growth and etc. manipulation for this agrobacterium. All the work I presented today was done by two truly amazing graduate students, Zhongqing Ren and Qin Liao. I received funding from Indiana University and NIH, and thank you for being here.
Thank you so much, Dr. Wang. It's very amazing talk. So I got a question from the chat box saying, from Mu Chen Yuan, said, thank you, Dr. Wang, for sharing. Are multiple chromosomes in bacteria usually of the same kind, meaning they are redundant in genetic information, or are they usually different from each other?
So the different chromosomes. Okay, first there is a distinction between a chromosome and a plasmid. If a molecule, so in our case, we have 4 different molecules. Why do we call this chromosome? This the others a plasmid. If a molecule contains essential genes, then this is meaning that the cells cannot live without this. Then this molecule is a chromosome. If you can delete any gene on one replicon and the cells are fine, so these are plasmids. So chromosomes contain essential genes. So, and it means that these they can't be removed. Usually for multipartite genome, the chromosomes contain different genes. So they are not redundant. They contain essential genes.
Cool, thank you. And her second question is a multipartite bacteria have multiple chromosomes from fission, or they are usually gained from the environment following stimuli?
This is a very, very good question, and people have been studying this for a long time. And at the beginning, there were a lot of debate whether it's one chromosome being split into multiple ones, or are there different molecules fused to different ones, to fewer ones. So for now, the consensus is different replicons. Different replicons, the secondary chromosomes are from plasmids. So at the beginning, maybe there is only one chromosome, then there is a plasmid, maybe the plasmid provides some useful things, and this plasmid is maintained quite stably. And then slowly this plasmid acquired essential genes. At the beginning, they can maybe during evolution, if you lose it, it doesn't matter. But later, when this plasmid now acquired essential genes, it become essential and now eventually become a chromosome. So the consensus is the origin of secondary chromosomes are from plasmid, and after they acquire essential genes, they become a chromosome.
Great, thank you. And I have a question of my own. So I'm wondering, like, see in this bacteria, the chromosomes are grouped together. I'm just wondering, is this grouped up chromosomes are actually important for pathogenesis or something? Do they have some actual function or something like that?
Yeah, it's a very interesting question. So the grouping of the chromosomes, we found that the major thing is if we can, when we disrupt this grouping, the secondary replicons get lost. So even in one of the slides, I didn't point out when we remove the partitioning protein PARP1 from the genome, the smaller plasmid get lost. So we think the grouping is helping the maintenance of these smaller replicons. These smaller replicons might not be useful in every cell generation, but they might be useful in other cases. So they've been evolved millions of years, so they must be useful for some cases. And having them grouped together with the largest chromosome help the maintenance of these smaller replicons. We haven't found, so because one of the plasmid is truly for the pathogenesis of this plant pathogen. So Agrobacterium is a plant pathogen, and now people use it to engineer plant cells. The smallest plasmid can be transferred into the plant, and it causes tumor in the plant. But researchers use this as a vector to change the plant genome. And we found that without the grouping, this small pathogenic plasmid get lost. So we think it's the one major importance for the grouping is the maintenance of small things. The other one is it might also play some regulatory role for the replication of these smaller replicons.
Very interesting. Thank you. And my second question is like, for the dicentric chromosome, do you have any direct evidence for it? Because do you see it actually under the microscope? Like the two chromosomes are actually fused, like what you showed here.
So I wish we have a technique to look at the DNA and be able to separate one chromosome from the other. So when we look at under the microscope, at least for now, we see the DNA, we can label specific region and look at the dots, but we really can't say that this blob of DNA belong to this chromosome, this blob belong to the other chromosome. But we do have evidence that these two, there is a fusion. So, and this fusion is at a specific location. So we found that on chromosome 1 and the chromosome 2, there is a very small region. It's kind of redundant. So they are very similar in sequence and differ by only 11 SNPs. On the original strain, we see this kind of sequence distribution. And in our fusion strain, we sequence these regions, we found that there is a recombination. So indeed, it is, there is, under the exchange is not a clean cut and rejoin. So there is one small region is identical. So this is indication that very likely there is a there is a DNA break, and then to repair the break, the cell use the other template. So in bacteria, they are really minimalist, meaning that they have only one copy of the genome. If there is a break in the genome, if there is no repair, the cells will be dead. But in this case, maybe it was lucky, it's broken at this place. And then it happened that on the other chromosome, there is a similar region, and they did a recombination to repair the break. But then it generated this weird chromosome. And it happened that the cell has the machinery to resolve this issue, and the cells survived. I think this is just a glimpse of evolution, and we captured an intermediate. It's really luck, I think. But in the wild, maybe this is happening all the time. And if the cells are not lucky, they will be dead, eliminated from the population. In our case, it's really luck that we found this. Yeah.
I see. Thank you. So another question from Jiang Xinhe, and it's in Chinese. So she said, 感谢王老师的分享，想请教 fusion 的情况是不是可以类推，也就是说，实际上不同 chromosome 或者 plasmid 之间的 fusion 可能是普遍存在的情况，还是您认为稳定和普遍存在的 fusion 是少数情况，需要满足一些特定条件呢？谢谢。
It's a very interesting question. So, in fact, it's the fusion between two replicons and the later split is quite common. It depends on the species. In one species called Xindan, it has 3 plasmids, one chromosome, 3 plasmids. And those different replicants, those especially the different plasmids, they frequently get fused and split very, very often. And in model species, when people study Vibrio cholerae, they have 2 circular chromosomes. And when you manipulate one chromosome, making it not segregate well, usually the suppressor is the 2 chromosomes will fuse. so that the second chromosome, it contains essential genes. They don't want to lose it. So they get fused to the large chromosome to ride on the large chromosome to survive. So fusion and splitting is very common, and I feel it's constantly happening in nature. And this is a part of the evolution. And whether we can capture this small stage or not is another question. But it's a very interesting question.
Great, thanks. So next question from Yunqing Wang. She said, in the XERC and XERD depletion mutant lines, did the chromosome topology actually change?
The topology of the chromosome doesn't change that much. What is really changing is at the cell level. The cells are really screwed up. So basically, the chromosome cannot complete segregation and block cell division. The cells smallly grow into a large cluster, all connected to each other. Eventually, those cells will be dead. Then, if we look at the high C, the chromosome conformation, the overall conformation doesn't really change.
Great, thanks. So next question from Jian Guo. She asked, would DNA tracing technique help with chromosome imaging, though it can only show several foci instead of the whole chromosome?
So it is very useful, and we do use it. But each time we are looking at only specific locations, it's a little hard to deduce the entire genome. Ideally, we want to have this chromosome painting technology when you can label many regions on each chromosome. So I feel this technique is for bacteria will be there in the future. Right now, we don't have it yet. It's a very interesting point.
Great, thank you. So I got last question here. It's from Liu Zhiyong, and he asked in Chinese, and it's about the personal development. So he said, 感谢王老师的分享，再次祝贺您 tenure 成功。假如现在让您重新 set up 一个实验室，和6年前相比，您觉得您有哪些东西还会和以前一样坚持？还同时有哪些东西您会觉得会 do something different?
This is a very interesting question. I think about this a lot. I feel even now, so I will answer your question a bit. So even now, when I look at what am I going to do in the next 6 years, right? I have a vague direction, but the specific topics or specific research direction, I feel is all decided by my students. So now looking back 6 years ago, if I start right now, I feel I'm going to still do very similar things. But I really can't tell whether I will be exactly the same or not in 6 years as I am now. It is because I will have different people in my lab, and they might have taken me to a completely different direction. So I feel there is a lot of random chance in there. Eventually, I would still be doing research, but the exact research topic will be changing a lot, really depending on what people I get, what they discovered, and what to do next. A lot of decision making to switch us one way or the other. That's what I think.
Great, thank you. So if no more questions, I guess that's it for today. 再一次非常感谢王教授的支持以及各位的参与。那么我们本期的学术讲座到此结束，谢谢大家。
好，谢谢大家。
那么之后呢，其实王教授也正在招聘他的这个博士生或者是博士后，大家感兴趣的话，我们之后会在我们的公众号里 也发一则这个招聘信息及广告，大家有兴趣的可以继续 follow 我们的情况。
对，如果有人愿意读博士或者做博士后，可以跟我联系，谢谢。
好的。那么王教授的联系信息呢，我们也会放到接下来推送里面。那么今天就到此结束，非常谢谢各位。
好，
拜。
谢谢王老师，拜拜。
谢谢。